cross-linked effector functions and lower cross-linked effector functions. We have been demonstrating that. Also, I want to know how IgM dynamics change in different species of fish. So other fish, in addition to the tetramer, trimer, dimer, monomer, also have intermediate species. And I'm curious how these intermediate species change throughout the immune response. I want to know the effects of introducing different pathogens. So in my case, I use bacteria. But what happens if we infect with a parasite? In mammals, IgE would increase for a parasite. So maybe instead of the tetramer, maybe the trimer predominates with a parasite. I want to know the IgM dynamics following natural routes of exposure. So rather than using a vaccine that has adjuvant, the adjuvant could have affected some of my samples. So I told you about that week 2 outlier in the total antibody proportions. That could have been due to an adjuvant. And we wouldn't see that with a natural infection. Also, I want to know how quantity, quality, specificity to protection relate when these increases don't coincide. So in my case, I saw them pretty much all increase at weeks 5 post-immunization. But are low amounts of highly cross-linked antibody better protect cheaper fish? So finally, I just want to get to the slide I started my presentation with. So what happens when we look at fish as unique organisms and not as mammals? What we see is that following vaccination, there's an increase in titer, increase in the specificity. Fish don't class switch. They don't go from IgM to IgG. But it seems they do IgM type switch, where you go from that low cross-link, low affinity, highly flexible antibody to the highly cross-linked, highly affinity antibody. They do experience affinity maturation. We saw that. And they don't rely on the classical form of memory, but they do rely on humoral memory. And I didn't demonstrate that myself, but Dr. Bromwich's previous research has suggested this. So I'd just like a few acknowledgments. And thank you.
We found someone that can talk more than me. Very effective way of not getting a lot of questions. So are there some questions for Greg now, please?
Yes.
We're going through your results. I noticed that for part of it, when you were comparing the specific response to the total response, you only really looked at tetramers?
Yeah.
So I was wondering why you didn't look at the other guys?
All right. That's actually pretty easy to explain. I mean, I went over this with Dr. Bromwich, actually. I figured somebody would ask that. Okay, so let's look at these different antibodies. Now, the tetramer can only be produced from a single structure. The dimer and monomer, in contrast, can be produced from many different cross-linked structures. So I can't really correlate what structure was responsible for producing a dimer and monomer. So no cross-linking at all would produce 4 monomers. Even this structure here, that has 2 disulfide bonds, is going to produce a monomer. So I can't tease that apart from my analysis. So that's a simple explanation. But also previous research has focused on the fully cross-linked antibody. Yeah?
This is probably going to be a difficult question to ask and answer, but have you looked through the majority of the monomers and looked at like the actual DNA to see if there are mutations away from cysteines, towards the binding areas, down at the bottom of the constant regions?
I haven't gotten into molecular at all. That's, I haven't.
The constant is very, very constant. There's only one that can come in. So the affinity changes have to happen in the top part, and then the structural changes can actually affect the head.
Well, I mean, like, is there like a breakdown of the cysteine preventing the sulfide bond to affect the constant?
What do you think affects the disulfide bonding pattern? What have you read about that?
What do I think affects the disulfide bond?
How does it happen? What are the biological mechanisms by which it could happen?
It's from I don't remember the actual pathway, but it's actually within the cell, the mannose, right, and all of that. And so it's actually the biological pathway within the cell that's causing that following binding. I don't remember the specific pathway, though.
You look at the sugar.
The sugar residues, yeah. else. Jeff?
Was there any evidence of a secondary infection in any of your fish? I'm just thinking about that one anomalous point, and maybe it could be a result of a secondary infection. That was only based on your gels, right? So it wasn't specific. It wasn't an Eliza or Western. So maybe a secondary infection would have introduced more low affinity antibodies and shifted a couple of the fish down.
That is possible because what our results are showing is that if you inject fish with 2 pathogens, then they actually are going to have to shift the response from just one to both of those pathogens. So that could have an effect if they did have. But within the fish, I didn't see any secondary infection or anything like that. So I don't think that's the case. I think it is an adjuvant effect.
We have time for one more question. There's one more here. And there's not. So thank you very much, Greg.